we're going to talk now about stem cell therapies, real possibility or not. And this will be presented by Dr. Ramona Doyle, who comes to us from California, UCSF, and a company called Blade. She's going to tell us more about this.
Thank you. First, I want to thank the organizers and say what a great honor and privilege it is for me to speak today about a topic fairly near and dear to my heart. So just in terms of disclosures, I think my new slides didn't make it, did they? Okay, well, I'll give the old talk. The slides were changed up, but I guess. Do you guys have the new slides? No? Yeah, okay, never mind. I'm just going to wing it. There's no CME credit for mine anyway. So first of all, I would just say that in the realm of speaking, I was burdened with a very terrible title, which I changed, but you won't know about that. So moving right along. In terms of disclosures, I am currently CMO of a small biotech called Blade. I have no financial interest in or benefit from stem cell therapies. I actually spent time at the California Stem Cell Institute, which is where I learned about stem cells. And unfortunately, my slides, one of the things I wanted to share with you is that that institute still has funding opportunities for academics and small companies who are interested in utilizing cell-based therapies. And if anybody has any questions about that, please see me later. So one of the first things, and I also want to thank and congratulate Sonia Danoff and Marilyn Glassberg, who I think have educated this audience and this community of patients and providers and researchers. I think that, I actually believe in science, and I believe in data, and I believe that we have to educate ourselves so that we can educate our patients and those in the community. And there's a lot of misdirection in terms of what's what, but I want to talk a little bit about stem cells, the term stem cells. First to say, stem cells, some of the promise of stem cells has already been realized, and thanks to the previous speakers, we understand that there are different varieties of stem cells. Hematopoietic stem cells are the cells that are progenitors of things that come out of the bone marrow, fundamentally. And stem cell transplantation, bone marrow transplantation, has been going on since the 1950s. And this is also so that everybody appreciates that there's a huge history. These didn't come out of the blue. All of this talk now is built on previous work. So in the 1950s is when stem cell transplants began. And at that time, we didn't actually understand enough about the immune system, but we were learning about HLA antigens. And the first bone marrow transplant was done after someone received a massive dose of radiation victims, thanks to what was going on in the world at the time. So in the 1960s, there were very important breakthroughs in immunology where we finally began to understand HLA typing in the immune system better. And actually the concept of an HLA-matched sibling transplant for severe combined immune deficiency syndrome. Now, everybody at that time, in order to make space in the bone marrow for the transplanted cells, you had to have a conditioning regimen, and we may come back to that. During the 1970s, there was a lot of experimenting around making that process safer and more effective. And over time, in the 80s, with registries and better matching and better understanding of the immune system and the complications of immune suppression, we saw better results. And basically what's been happening since the 1990s is we've discovered that you can actually extend indications beyond what I would say cancers, fundamentally blood cancers, where you can actually do genetic diseases. And I think this is really important when you think about what I would call the low-hanging fruit in stem cell therapy, and that would be patients with monogenic diseases who you can harvest a stem cell, you can CRISPR correct for the gene defect, and you can put it back in. And there are studies going on right now in patients with sickle cell, thalassemia. There have been cures. Don Cohn at UCLA in children with ADA SCID has shown with gene correction that you can actually cure this. The other thing that I'm not going to get to, but I wanted to talk about, is the advances in using viral vectors to bring things into situations where there can be correction with that vector. And just in the last few weeks, there's been data on correction of the SMA1 in children with spinal muscular atrophy, some of the eye diseases as well. So there's many ways beyond stem cell, what I would call cell-based therapies, where you can see benefit. Also, in terms of the whole history of it again, and we'll get to this, improving the sources of the stem cells. So I like to think of this broadly as regenerative medicine, which is really where we're bringing translational research into not just stem cells, but tissue engineering, molecular biology. It involves replacing, engineering, or regenerating human cells, tissues, or organs. And I would broadly say that some of the things I've mentioned have to do with regenerative medicine, and stem cells in particular come under that rubric. This is just to point out, and other people have pointed out, there's been a lot of development. Stem cell treatments did start in the 1990s, but importantly, it was shown, Dr. Yamanaka, who eventually went on to win the Nobel Prize, showed that you could reverse engineer pluripotent stem cells using a series of factors. And by the way, I have to give a nod to the folks who labored in developmental biology for many years to understand the programming. You know, we had to know how we got from A to Z as we went from from a blastocyst to a human being. And I think the people that painfully reconstructed all the signals and pathways were the ones to whom we owe a lot of this. So in terms of sources of stem cells, there are, we've talked about the bone marrow, and for reasons that are clear, there are certain diseases that may or may not be appropriate. Now, there are also diseases where you really want a stem cell line. For instance, in Parkinson's disease, there are absolutely stem cells that are neuronal stem cells that have been engineered to produce dopamine. And there's a lot of clinical trials going on right now. There are cells that have been engineered to produce insulin. So there are specific needs for specific cell lines. At CERM, a lot of the projects we had, we actually had many in eye disease. One of the reasons for that is that the eye is somewhat immune privileged, and the ability to reverse engineer retinal pigment epithelial cells and place them in the eye has made that an attractive target as well. So again, just to review just a little bit about definitions, because I am a somewhat retired professor. Embryonic stem cells, you've heard about all of the challenges with those. They come from the inner cell of a blastocyst, ergo from embryos, and there are lots of ethical and other concerns with that. But there's also the pluripotency means that there's the risk of teratogenicity and cancer, and that has been an ongoing concern, though people are meeting that concern. Hematopoietic stem cells is really what I started talking about. They're multipotent. They can form lymphoid and myeloid cells. There are cells that are progenitors of mesenchymal cells, which come from the bone marrow, and not necessarily from adipose tissue, but I won't belabor that. And their differentiation potential is different, but nonetheless. Mesenchymal stem cells I'm going to spend some time on because the one trial in stem cells and lung disease it uses this as a source, and Marilyn's talked about that a lot. You can get those stem cells from different sources, and I think we've done this particular meeting has done a good job of trying to put into perspective the stem cell tourism and what's real science and what's just bogus. And I think the PFF community is lucky to have advocates and scientists who are helping guide this group in terms of what's legitimate for patients in terms of research and clinical studies versus what is just simply stealing money from you, a la the snake oil approach to medicine. So in terms of the MSCs, there's a lot of things about them as a part-time drug developer that make them good in terms of production, and I'm going to talk a little bit about some of the challenges. They can be readily cryopreserved, they can be tumor trophic, and they can be readily genetically manipulated. They do have, depending on the way that you produce them, may have a limited yield. Induced stem cells, basically derived from somatic cells. You can reprogram a skin cell back to be pluripotent and then forward to be something else. It's kind of magic. So you can drive into many different cell types. You can actually create patient-specific cell types. And there is some tumorigenicity potential heterogeneity is a problem. I will just say that CIRM, one of the things that the California Stem Cell Institute has, and is not, I think, being used, is they have a repository of induced pluripotent stem cells from 300 well-characterized patients with IPF that are ripe for study if anybody here is interested in pursuing that. So a lot of the themes in terms of regenerative medicine, there's everything from regulatory challenges and production challenges to just sheer biology and scientific problems. We've talked about whether something is from you or from someone else that can pose an issue with the immune system depending on the cell types. As Marilyn said earlier, MSCs don't have MH2 complexes. They generally aren't a problem. So the idea from a stem cell clinic that if they're your own cells, they're better is really a vast oversimplification of a lot of complex biology. But in fact, if we go back to the hematopoietic stem cells, we know that it's better to get your cells from a matched sibling than from a total stranger because we understand now the various components of the immune system. I think the other thing is pluripotency. The ability to be a lot of things is a good thing. It can also be a bad thing. In terms of programming and how you program and then monitor cells for their teratogenic or tumorigenic potential is still an area under a lot of discussion. The other thing is that I think we are fundamentally dealing to a large extent with three-dimensional biology in two-dimensional spaces. We're relying on cell cultures when in fact, for instance, the survival of dopaminergic neuronal stem cells in the brain probably, why isn't that working? It's not. We can do it and we can put them in the brain, they make dopamine, but how do we know how long they live and what is the 3D configuration they're in and how important is that to their survival? So there's a lot of really interesting challenges there. In terms of just making these drugs available or these therapies available, there's a lot of issues around safety and efficacy. Which cells do you use? How do you upscale the production? How do you do it in a way that's cost efficient so that every patient who might benefit actually can receive these therapies when we get around to it? And to a large extent, you know, in the old-fashioned world, you know, making drugs was chemistry. And now, in terms of cells and biologic therapy, the process is the product. And every time you improve the process, maybe make the better yield for the cells, et cetera, you may make a change that then can impact whether or not the regulatory authorities are going to feel that what's come out at the other end is the same as what you started with. And these are considerations which can cause a lot of expense, and so it's important to think about. Just a quick word about what I call the yin and the yang of MSCs. MSCs kind of, we don't actually know how they work, and I would say that it comes down to this conversation for me. Are they drugs or are they drug stores? Marilyn's already told us you inject MSCs into people, they're in the lungs within 24 hours, they're gone. So what are they doing? They don't stay there. They're probably dropping off potentially packets of good things that we don't understand. I would challenge the community to let's begin to understand what it is about, what is in the mix of mesenchymal stem cells appropriately processed and characterized, not from your fat tissue. What is it about that we think might be transformative for patients with lung disease? In terms of clinical benefit, I think depending on how sick patients are, they might be willing to tolerate more invasive regimens. I know that, for instance, in sickle cell disease, where we're trying to do corrective gene therapy, where you do an ablation of the bone marrow, kind of a bone marrow conditioning light, doing that in a patient with sickle cell on the chance that we can give them a gene-engineered corrected red blood cell progenitor is a little scary. But again, it depends on where the patients are. For a child who's going to die of spinal muscular atrophy or an adult who's going blind, the risk-benefit in terms of the conditioning regimen, they might be willing to take on more. I think the other challenges in cell-based therapies are just how do we know that the cells that we have made and do what they do are alive and continuing to do what we need them to do, and how do we ensure they're in an environment, a 3D environment that keeps those cells viable? I've mentioned immune suppression and tumorigenicity. And I want to get to the lungs here in the last few minutes and just say that it's not just one size fits all. Depending on where you think the keys, where anatomically and the relevant activity is for the disease in the lung, and particularly in an IPF, where you look at which stem cells, which progenitor cells is very important, and it varies. And you can see that in the large airways, you can see across there some of the related lung diseases. We think about cystic fibrosis and COPD. As you go down that tree, pulmonary hypertension, which involves the blood vessels, of course, ARDS, acute lung injury, and I should say that I've had the pleasure of being mentored by and working with Mike Maffei, who's also doing a trial in acute lung injury in patients on ventilators. And also then further down into the lungs, I think is where we talk about where a lot of the action is for IPF and for bronchopulmonary dysplasia, which is a terrible lung disease that affects children. I just want to let you know that there are actually a lot of clinical trials in lung disease using stem cells. I won't go into the details, but there is one of the things that CIRM did is we did a workshop on regenerative medicine in the lung, and many people in the community attended, Mike Maffei, Bruce Trapnell, Marilyn, and several other folks, and that publication just came out and is I think a good scientific summary of the state of the state in stem cells. In terms of the providing possibility and hope, I just want you to know that I approached my work as I joined the Stem Cell Institute for the year I was there with a great deal of skepticism. And I actually now really believe that with good science and good technology, we're going to get there. But we can't skip any of these steps. And the other thing I want to say is that sometimes it feels like the FDA is getting in the way of progress, but their job is to protect folks from, their first job is to keep things safe, and we're very fortunate to have that type of regulatory authority. While it may be frustrating, I think, for us sometimes to deal with the regulatory rules, they're there for a reason. And so I think right now with stem cell therapies, whereas with other therapies we sometimes get frustrated by the long and expensive course to get a drug or a therapy approved, I think with stem cells, they've got this right, and they're looking out after folks. The way I think of it right now is a quotation by an author who I love, Flannery O'Connor. It seems appropriate, I'm in the South. Flannery O'Connor was asked whether she thought that these creative writing programs, aka the FDA, were stifling too many good, creative, innovative writers. And she thought for a minute. She says, I don't think they're stifling enough of them. And I think right now with stem cell clinics, that's how I feel. And I think the FDA actually needs to do more, not less, to get in between vulnerable public and bad, not science. I'm not going to say fake news. Finally, I did it. I've already said it. Finally, I want to talk about, I've talked a little bit about the low-hanging fruit disease-wise. Now, I'm a pulmonologist, so I am anxious to push forward in lung diseases. But let me also talk about how stem cell and cell-based therapies can make therapies more readily available by moving them through this development path quicker and with less cost. So stem cells, there's some great pioneers in this. Joe Wu, who's a cardiologist at Stanford, has done great work with taking cells from patients who have complex arrhythmias, putting them in and reverse engineering them to the patient's own cardiac cells and showing which combination of antiarrhythmic drugs can prevent lethal arrhythmias. This is actually being done. That's an example, and there are other cell-based systems which can make the innovations that we need in drug development more readily available to all. So I think, in summary, I would say that cell-based therapies are already here. They've been here for a long time. This is just an amazing period of time with technology. I think technology is actually going to solve a lot of these problems. But honestly, there's no quick fix. Good, hard, dogged science is what we need, and we need patients and providers and folks in the community to understand and appreciate all the many big shoulders that we all stand on. And that, again, I think we have to and I know this organization and many of the people in it stand up for science and for data, and most importantly for patients. And I feel that the future is bright, and I believe in it, and I'm very honored to be here today with with all the speakers here, because these are the folks who are going to deliver on the promise of new therapies. I really believe it. Thank you very much again for the invitation to speak here. Thank you.